Now, the kidneys filter approximately 810 millimoles of potassium per day. However, the average person has a total extracellular fluid potassium of about 63 millimoles. At this rate, the kidneys would deplete the extracellular fluid potassium in about 2 hours. So it clearly suggests that the kidneys reabsorb a large portion of the filtered potassium load. In fact, the kidneys reabsorb 95% of the filtered potassium load, or 770 of the 810 millimoles that are filtered. Well, 80% of the filtered potassium load is reabsorbed along the early and late proximal convoluted tubule. 10% is reabsorbed along the thick ascending limb. 2% is reabsorbed along the initial and cortical collecting duct via alpha intercalated cells, and 6% is reabsorbed along the medullary collecting duct via alpha intercalated cells. Now, this means that approximately 2% of the filtered potassium load is excreted by the kidneys. So let's ask this question. Now, reabsorption of filtered potassium along the proximal convoluted tubule occurs paracellularly, driven by solvent drag in the early portion and a lumen positive potential in the late portion. Now, solvent drag accounts for most of the potassium reabsorption along the proximal convoluted tubule. And as a reminder, solvent drag along this nephron segment is dependent upon sodium reabsorption. Reabsorption of potassium along the thick ascending limb occurs equally between transcellular and paracellular pathways. Now, the transcellular reabsorption of potassium occurs via the sodium-potassium 2-chloride cotransporter. The apical potassium channels serve to recycle potassium, and without this source of potassium, the sodium-potassium 2-chloride cotransporter would cease to function, or function at a lower rate. Now, paracellular reabsorption along the thick ascending limb is driven by a lumen positive potential, which is created by the differences in potassium and chloride permeability between the apical and basolateral membranes. Now, reabsorption of filtered potassium along the initial and cortical collecting duct occurs exclusively by alpha intercalated cells via a transcellular pathway. Now, transcellular reabsorption of filtered potassium occurs via the apical hydrogen-potassium ATPase, which transports one potassium ion into the cell in exchange for one hydrogen ion. This process requires energy in the form of ATP. Well, reabsorption of potassium along the medullary collecting duct occurs via paracellular and transcellular pathways. It also depends upon the upstream secretion of potassium by collecting duct principal cells. Principal cells secrete potassium into the lumen via apical potassium channels and concentrate the luminal potassium by reabsorbing water via water channels. Now, the increased luminal potassium passes into the medullary collecting duct, where it favors passive reabsorption via paracellular pathways and active reabsorption via the hydrogen-potassium ATPase. In addition, the reabsorption of luminal potassium along the medullary collecting duct contributes towards the recycling of potassium between the descending thin limb and thick ascending limb. And finally, as we mentioned earlier, under normal extracellular fluid potassium concentration, about 2% of the filtered potassium load is excreted by the kidneys, which means that of the 810 millimoles filtered, 16.5 millimoles will be excreted per day.